what can the genomics revolution do to help patients with chronic aspergillosis? And that might sound pretty off the wall at first. So before I get into how it applies to aspergillosis, I'm just going to just explain what I mean by this. I used to work at a different hospital down in Birmingham. There was the genetic testing centre, like for the whole region. So I used to work there, and it was really interesting. And we're in a really, really exciting point in history in terms of genetics, because we've gone from like pretty much no genetic testing to having all these amazing, like really fancy high throughput technologies that do all sorts of amazing things. So the NHS obviously has a PDF document explaining all the strategy, as they do for everything. So the quote that I thought was really nice says, "Genomics will be at the heart of this future and the next generation of healthcare in the NHS." So this is a plan for how to take this technology and apply it to what challenges we're actually facing in the NHS. So when people think about genetics, the first thing that usually comes to mind is inheriting diseases. So things like cystic fibrosis, which is a predisposer for ABPA, that is kind of what most people think of first, but it's not just about that. So 50% of the service that I worked at, it was about cancer, because cancer cells, they're genetically very, very weird, and I'll explain in a minute how the testing helps for that. There's also prenatal testing, so things like Down syndrome is the classic one. So that's not something that's passed on from a parent, it's something that just happens for that child. There's something called precision medicine, which I'll explain in a minute. And kind of late to the party, infections. It's not what the genetic testing centres normally work on, but what we can do is use the technologies that they're spearheading and apply it to more and more things. So all different types of medicine. It'll sort of spread throughout all the different types and be properly integrated. So back in, I can't believe it's 2018 now, that's a long time ago now, they ran something called the 100,000 Genomes Project. And the idea of that was to find out what people are like on average with their genes. So they got 100,000 people and they sequenced all of their genes, just the whole lot. And that's really good. Basically that's going to be our control groups for loads of stuff. They avoid talking about people as being normal, because there's with genetics there's there's just a lot of variation, and it can be quite difficult to say what actually normal is. There's just a lot of variation. And so yeah, there's various different roadmaps for how to get this spread right across the country to hopefully benefit as many people as possible. So with the cancer side of things, this is actually really, really starting to get turbocharged. If anybody knows the Christie, the hospital in Manchester, the stuff they do there is just wow. So there's some of the genetics is starting to become pretty widespread and starting to become a bit more known by the public. So with cancer cells, there's no such thing as a genetically normal cancer cell. Every single cancer cell has things like genetic mutations, and that's what drives the growth. So some of those mutations can be inherited. So if you've heard of BRCA1, that's a breast cancer risk factor. So it doesn't, if you inherit it, it doesn't guarantee you'll get breast cancer, but it makes your risk much more likely. So that's what Angelina Jolie, I think she had a preventative mastectomy, I think, because she was carrying that gene, and that's a very personal choice what you want to do in that situation. But also there's quite a lot of genes, they arise, they call it somatically. Soma just means body, like that's all that means. So it means after you're born, then for some reason, like maybe sort of exposure to some sort of toxins, or well, lots of reasons really. Just when the cell divides, it just makes a little mistake, but that happens that's after you're born. So there's lots of different ones, and they also have, when you look at them under a microscope, they have really abnormal chromosomes. They're all broken up. There might be too many of one and none of a different one. It's usually very clear under a microscope which ones are cancer cells. So in the picture there, on the left, that's what a usual cell would look like. So 23 pairs. So you've heard 23andMe. That's because there's 23 different chromosomes, and you've got 2 of each one, because you got one from your mum and one for your dad. And they when the cell divides, they're supposed to go into their pairs and line up nicely and then be separated. But with cancer cells, the whole process is just gone wrong. It's a mess. And yeah, you can so the picture on the left, that's just one, it's called a karyotype, when you see all the chromosomes laid out. So you can see how different that is to what it should look like. And the reason this is important is because when you know the mutations, some of them, it tells you something about the biology of the cancer and how it's going to behave with different drugs. So the really famous one is Gleevec. It's called, like, Gleevec's the brand name, and imatinib is just the chemical name. That's used for a type of leukemia called CML. And when they introduced that, the 5-year survival rate, it used to be about 30%. So like 1 in 3 people would still be alive after 5 years. And just with that one drug, it jumped up to 90%. And that is just monumental. So it can be a really powerful tool. There's also another side of genetics is precision medicine. So the reason why this is, it's a slightly different way of looking at things. So most clinical trials, I think people generally understand that when you've got a clinical trial, it's better for it to be on a big group of people to make it more reliable. But something that we don't always talk about is, within the group of people in the trial, are they all quite similar to each other? Or have they got different subtypes of the disease, or they developed it for different reasons? And I think with aspergillosis, obviously, we're used to the idea that when you say aspergillosis, you can mean lots of different things. So that's called stratification. Which stratification just means like layers, like there's different, within your group, there's different kinds, basically. So the best way I've ever heard of describing this, and it sticks in your mind, is what's the average number of boobs per person in the UK? So if you just took an average, like the number of people divided by the number of boobs divided by the number of people times 100, you'd come out with a number about 1. And yeah, it's numerically it's the average, but it doesn't tell you anything about reality. It's that we're not a nation full of people that all have 1. Either people have some people have 1, but most people have none, or they have 2. There's so that's what I mean by stratification. So some new treatments work better if you can work out which type of patient people are. You can target, if you've got different medications, you can target the right drug to the right patient and get better outcomes, basically. And that is called pharmacogenetics, which is an unnecessarily long word for it, I think. But it's very, very exciting. And there's a lot of applications. So I'm not going to go into all the different kinds because it's lots of different diseases, but you can use it for things, I think, arthritis, whether you qualify for biologics or not. I think they're already starting to use the genetic tests to basically to see whether it's going to work or not. Because some people, you could give them a treatment and nothing happens, but they still get all the side effects. And another example that I've seen at a conference is heparin dosing. So making sure you give the person the right amount and it's going to get up to the right level in their blood to begin straight away. So like I said, kind of late to the party. Infections, then you wouldn't have that tested for at the genetics centre, but different laboratories around the world, around the country, are bringing in more and more tests in sort of microbiology labs. So what you can do with this is each different type of germ, like every virus or bacterium or fungi, within their DNA, they all have unique sequences. And if you want to know who's there, you can sequence that DNA, and it's like a little barcode. So you read the barcode and you compare it to a database, and it can tell you what species it is, or sometimes even like what strain it is. So if there's an outbreak of something, it can tell you, yeah, this matches, these ones all match, these are all from a common source. There's different, it all gets, obviously everything gets complicated once it hits the real world, but if you're interested in reading about this, there's one called ITS, that sometimes it's called the universal barcode, because it's in bacteria, it's in plants, it's in humans, and there's lots and lots is known about it. So it's very, very useful. There's you will probably have heard of PCR because of COVID. So that's been around for quite a few years. It's not a sequencing test. The way that works is you type out a little sequence that you want to look for, and then the test says, is it in this sample or not? And sometimes how much is in there if you're doing certain if you're doing it fancy. So PCR is more of a, is this there, yes or no? And that's something that's in all hospitals now. And you can even get little home machines to do a very basic PCR. I actually know one of my friends has one at home that she bought online. So I need to have a look at that, and it's quite interesting. So that's been around for a while, but the more recent technologies, they call it high throughput, and they are not kidding when they say high throughput. It is orders of magnitude more data, more different sequences, more different genes all at once. So not only is that cheaper, so more places can buy the machines, but it lets you do more other types of tests. So if you have a patient and you're not sure what is going on with them, say they've got pneumonia and you don't know which organism is causing it, you can take some sample from their lungs and test it against what you call a gene panel, which basically just means run quite a few tests at once. So there might be 5, 10, 20 tests, all just with one little sample, which is really good. And Another thing that this lets you do is you can, in some cases, you can measure how much of the pathogen is present. So that's something that we're interested in at NAC. So if somebody has like a long-term Aspergillus condition, you might find that somebody's they call it being colonized with Aspergillus, which means there's some growing in there, but not enough to cause disease. But if you measure that person over time, if you see that the amount of Aspergillus is starting to creep up, that might mean that they're about to have a relapse and they need to go back on antifungals. So that's a very useful one. So if anybody sends off sputum samples, that's something they quite often do for that. Something else really very cool is they can diagnose mixed infections. So especially with longer term things, you tend not to get just one germ by itself. They form horrible little communities quite often where they help each other out. And with old-fashioned testing, you couldn't detect that at all. But with the high throughput technologies, you get like almost like better resolution on a camera. You can just see more, and you can see, oh, this is Aspergillus and mucor, for example. That's very, very, very useful. And also you can, depending on what sequences you're looking for, you can look for resistance genes. And that can be quite handy, because so traditionally we would test for resistance by growing Aspergillus from the sample sent in and then testing it against all the different antifungals at different concentrations. So it's very, very labour intensive. Not many centres do that kind of testing, and also if it doesn't grow properly, you can't run the resistance test, the susceptibility testing. But if you can if you can find the genes directly, that can sometimes give you more information and choose a more effective treatment. So how does this apply to aspergillosis? So MRCM, which is NAC's mycology reference lab. So it's a reference lab, which means it's not like a normal hospital lab. It's like a regional. So we're in Manchester, and we do most of like the northwest of the country, and some specialist tests. We offer them all over the country, in like other hospitals, and also sometimes non-NHS clients. So if you're a professional and that sounds good, if you want to send your sample somewhere, I've put the website there, and there's a test catalogue, and it tells you the prices and turnaround times and everything, and they're very good. So with aspergillosis, where are we headed next with DNA-based technologies? So there's several different applications, and they're really quite separate from one another in terms of what they're doing. So the first one we're going to talk about this morning with Paul Bowyer, and then also Graham's going to talk about a different side of it, is detecting risk factors. So looking at genes involved in the immune system that can make you more susceptible to growing Aspergillus and getting ill from it. Another side of it that I'm going to talk about is how we can look at genes that control how you respond to voriconazole to avoid toxicity, and whether it's worth doing the genetic testing. And another one that we're not going to get too much into today, because there's a lot of other groups that already do a really good job of talking about them, which is antifungal resistance and surveillance. So that is so a centre like NAC will keep a big database of all the different resistance that we've seen, and then we can compare that over time to see what changes are happening, and we can compare it with other centres to see if different things are happening in different countries, and that's called surveillance, like just keeping tabs on the resistance and how things are spreading or changing over time. So those are quite different, quite different sides of it. And all of them have become possible because this genomics revolution has made the equipment and the technology much more common, more accessible. And now we can start to apply it to aspergillosis.